Hello everyone. Today we will look at another aspect of cellular response to injuries, cell death. Different from degeneration, cell death is the consequence of irreversible cell injury. Below are the major objectives for you. Define and recognize necrosis. Describe morphological appearance of different types of necrosis. Analyze the consequences of necrosis and compare necrosis and apoptosis. As always, we will first share a case. A 45-year-old man came with a 1-month history of low-grade intermittent fever and weight loss. He also complained of a painful swelling in his right axillary region. The swelling had been gradually increasing to its present size. These two pictures revealed growth and histological appearance of his axillary lymph nodes. What kind of necrosis does the lymph nodes show? I hope you will be able to answer the question after we complete this session. Necrosis is the localized death of cells or tissues occurring in the living body. Localized and living body are important points in this definition. If all the body cells have died, then we have a dead person, not necrosis. Necrosis occurs when trauma is so severe that cells cannot maintain homeostasis or adapt to the injurious stress caused by the etiologic agent. Necrotic cells are unable to maintain membrane integrity, and their contents often escape. Lysosome rupture leads to autolysis. Cellular contents leak out, resulting in inflammatory reaction and heterolysis. Basic morphological features of necrosis can be recognized under light microscope several hours after functional cell death has occurred. Before structural changes, cytoplasmic enzymes of dead cells have already leaked out into the bloodstream, and some of these form of the basis of diagnostic laboratory tests. The main morphological change of cell necrosis is nuclear changes. They are all due to breakdown of DNA and chromatin. There are 3 patterns of nuclear changes, including pycnosis, which is the condensation of chromatin and shrinkage of the nucleus. Karyorekesis refers to the fragmentation of the nucleus, while karyolysis means dissolution of the nucleus. Let's look at these photos. A shows normal liver cells. B depicts pycnosis with nuclear shrinkage and increased basophily. The DNA condenses into a solid, shrunken mass. C is karyorekesis, where the pycnotic nucleus has undergone fragmentation. In 1 to 2 days, the nucleus in a dead cell completely disappears. That is karyolysis, shown in photo D. For a pathologist to diagnose cell death, the key point is to look at the cell nuclear morphology. The sequence of the nuclear changes varies. A normal cell can undergo karyolysis directly or starts with pycnosis or karyorekesis. Besides nuclear changes, there are changes in the cytoplasm and interstitial tissue. Changes in cytoplasmic staining are also characteristic. Cytoplasm shows increased eosinophilia due to enhanced binding of eosin to denatured cytoplasmic proteins and loss of RNA. We can see a more glassy homogeneous cytoplasmic appearance than viable cells, mostly because of the loss of glycogen. Rupture of the necrotic cell membranes causes cellular contents to leak out, inducing inflammatory reaction. Breakdown and liquefaction of interstitial tissue produce a sheet of obscure, granular, unstructured eosinophilic material. Here, I would like to ask you a question. How can you distinguish necrosis from postmortem events? Cell necrosis in living organisms almost always initiates a local inflammatory reaction. Autolysis is the non-localized digestion of cells by enzymes in older cells of a dead body. This is a generalized event that occurs in normal tissues in a dead body. It is distinguished from necrosis by being diffuse and not associated with an inflammatory response. Now let's take a look at these two pictures. They are both liver tissue. Could you tell which one is necrosis? Well, this photo shows liver necrosis with inflammatory cell infiltration here. The other one is autolysis. Necrotic tissue can have several different appearances, which are sometimes helpful in making a diagnosis. Morphological changes of necrosis are associated with two factors: denaturation of cytoplasmic proteins and enzymatic digestion. Coagulative necrosis is a form of tissue necrosis in which the cells are dead, but the basic tissue architecture is preserved for at least several days. It might be due to the absence of proteolysis of the dead cells. Coagulative necrosis usually occurs in solid organs, such as heart, spleen, and kidney. Here, we have a coagulative necrosis of the kidney. You can see the wedge-shaped necrotic area. It is pale, swollen, and firm. The margin is defined by a dark red halo around the necrotic focus. That is hemorrhagic marginal zone. Under light microscope, the cell detail is lost, but the basic tissue architecture is preserved. We still can recognize the outlines of the renal tubules. This type of necrosis is frequently caused by lack of blood supply. Caseous necrosis is a special type of coagulative necrosis related to tuberculosis. The necrotic area contains more lipid, so it looks like cheese. Microscopically, the architecture of the tissue is completely destroyed. These pictures of tuberculosis case show the gross cheese-like necrotic tissue and microscopically caseous necrosis with unstructured and eosinophilic appearance. In liquefative necrosis, the necrotic tissue is completely digested and turns into viscous liquid. Discharge of the liquefied products leaves a cyst space behind. The liquefative necrosis can be seen here. We have tissues containing more lipids and little proteins, such as the brain, and tissues rich in enzymes, such as the pancreas. Abscesses caused by focal bacteria infection are also typical liquefative necrosis because bacteria stimulate the accumulation of inflammatory cells and the enzymes of leukocytes digest the tissue. Can you find the liquefative necrosis? Well, this is the liquefative necrosis with removal of some dead tissue. Microscopically, liquefative necrotic area appears as amorphous tissue. Now let's look at these two pictures again. The patient had symptoms of tuberculosis, such as low-grade intermittent fever and weight loss. On the cut surface of the enlarged lymph nodes, we can see the cyst-like necrosis and microscopically the unstructured necrotic tissue. Hence, this is the caseous necrosis of the lymph nodes. Let's summarize now. In this session, we have merely talked about the basic morphological features and patterns of necrosis. These include nuclear, cytoplasmic, and interstitial tissue changes. Among them, nuclear changes are most important. Coagulative and liquefative are two major types of necrosis, and they can be recognized under light microscope. Caseous necrosis is a special type of cell and tissue death. Next time, we will introduce more special types of necrosis and apoptosis. Here are the references and acknowledgement. Thank you.